Hello, I'm gonna look at some sun stuff. Not that. This one. We're continuing this paper that I was reading before called Engineering Strategies for Generating Hypoimmunogenic Cells with High Clinical and Commercial Value. So, SENS related, and I lost my place. Make it a bit bigger. Okay. Okay, I think this might be where I was. I think this is it. Alright. Ablate. What the fuck is ablating? Remove body tissues surgically. Gradually remove material from something. By melting, evaporation, frictional action, etc. Or erode in this. Remove by cutting or by erosion, melting, or evaporation. Right. So it's not actually very specific. I guess they mean surgical cutting. I don't know if they do. Why are we ablating the molecules? Okay, I don't really know what they're talking about so far. Okay, so one and two mismatch results in a cryotoxic CD8 T cell and CD response, respectively. Wait, mismatch with what? Because if they each have a response that's different, then they're not talking about mismatch with each other, we'd have one response. So they each have a mismatch with something, and then you got a response. Both ultimately leading to rejection. I think these are different type of mismatches you can have when you're introducing cells into the body. Genome editing platforms like CRISPR means the proteins can now relatively easily be eliminated. Oh, okay, so we're trying to get these proteins off of our molecules, I guess. So that's why we're ablating. We're trying to like evaporate them off the edges or something. Because these are probably the surface molecules that are used for identifying whether it's a body cell or not. Well, I'm not sure about that. I don't know if you have to have the correct ID on your molecules or if just like no ideas. Okay. Oh, abolition of HLA class 2 does not result in a missing self-response. So if you just get rid of it, you're okay. You don't get the autoimmune response. Only antigen presenting cells have HLA class 2, and if knocked out, it would render these cells non-functional anyway. Okay, so they might only have to remove one of the things, not both. to avoid autoimmune. That's what I'm getting. Oh, it says that up here too. Knockout of the ones is much greater importance because the ones are expressed on all nucleated cells and platelets, while the twos are only found on professional antigen-presenting cells such as macrophages, dendritic cells, and T cells, but can be expressed sometimes under other cells. I'll look up cytotoxic, came up again. Toxic to living cells, got it. And the bot showed up. Two significant problems. Natural killer cells can no longer identify them as self and so kill them. And what is called a missing self response. Oh, okay, you can get rid of the number 2 thing, without causing missing self. But if you get rid of the number 1 thing, then you get missing self. That's what I think they might be saying. The cells need identification, but they only need the type 1 identification, not the type 2. So for the type 2, the easiest thing to do is just get rid of it entirely, rather than make it work. That might be it. So what are they saying next? Groups engineering, class 1 cells, knock in one or more. Knock in, huh? Alright, well, I guess it means add it somehow, but usually you say knock out. So I don't know what that process is specifically. One or more immunomodulatory molecules to inhibit NK cell-mediated cytolysis. Dissolution or disruption of cells, especially by an external agent. Sure. Okay, so they add immunomodulatory molecules, meaning molecules that modulate the immune system, like control and regulate it. Exert a modifying or controlling influence on. Yeah, that. Okay, so instead of putting the correct identification on the cells, because that would have to be done on a per patient basis, what they're trying to do is put in cells that can control the natural killer stuff. Or not cells, but like pieces of cells, part of a cell. They add a thing to the cell that is able to like mind control the natural killers to not kill it or something. They don't have minds, that's like a analogy. Okay. They're presenting cancer and virus-derived antigens. Pivotal role in cancer immune surveillance, as well as the removal of virally infected cells. I don't know what cancer immune surveillance is or what it has to do with the topic. HLA-1 deficient cells may lose their predominant mechanism for doing this. Doing what? If they were better writers, it'd be easier. It matters more for people who are like struggling to read it. If an expert's reading this and he like already knows everything, then when they write badly, he's gonna be able to fill in the blanks or figure things out better. But the more someone's having a hard time reading it, the more the writing errors get in the way. That's why people need to write better when they're beginners and they're talking to me, because there's a big gap in our perspectives. Like, we're really different. So the writing errors cause more communication problems because we're already trying to communicate over a larger gap. So doing what though? Is it immune surveillance? Or is it something from a prior sentence? Not sure. Alright. That is a worst case, but presently only hypothetical. If they express the immunomodulatory molecules. Oh, if they became cancerous. They could be transmittable from one individual to another. Oh, you could have like cancer that that's infectious to other people, potentially. If you had these cancer molecules that were designed by us to not look like foreign bodies. So our immune system would leave them alone, right? So if we make cells that the immune system will leave alone, and then they turn into cancer, and then someone else gets them, their immune system would leave it alone, and then they'd have cancer. Okay, so that's a little bit scary as a worst case scenario. Given the absence of markers as which to be recognized as non-self and the presence of a shield to protect against a missing self-response. Okay, right. So they wouldn't have the identification, and they have what they're now calling it a shield as a metaphor that protects it against the natural killer thing that gets rid of foreign cells. It's not inconceivable that hypoimmunogenic allogenic cells for human use in clinical trials and beyond may need to be engineered with HLA class IA being left intact. A safety net in the form of a suicide switch or another analogous mechanism might be applied, which could be activated in the event of complications to attempt to clear administered cells, although such approaches remain largely unproven in this context. Okay, when they say if knocked out, it would render these cells non-functional anyway, it's not clear which cells they mean. That's hard to follow. You need context and like domain expertise just to follow the grammar, which is bad. It'd be better if you could tell what these cells referred to without being familiar with biology. But I'm pretty sure you can't. I think you just have to know the field. They were talking about multiple types of cells, like the natural killer cells. They could be talking about like knocking out the predator cells that are going to kill them, or they could be talking about it would make the cells they're adding to the body non-functional, rather than the immune system ones. I'm not really sure. Why did this? Why is it off center? What the fuck? Now it goes back. It's like left and right. Like it's like paper. It's from a fucking paper journal or something. And so they do the margins differently based on whether it's a left page or a right page. They want a larger margin in the center where the spine of the book is, because you lose more of the page to like the folding in the middle, kind of. So that's why they do that, but it's dumb to have that in the electronic version. Like the electronic version is screwed up based on the paper version. Hello, Alex. Okay, where was I? I was here. Alright. Engineering of immunomodulatory class 1B and immune checkpoint molecules. Okay, so before we were talking about 1A and 2, but now they're talking about 1B. So it's switched. Tolerance promoting molecules. So tolerance promoting means like it tolerates it in the body so the natural killer doesn't kill it. So you put those molecules on the surface of cells will help with hypoimmunogenicity, which just means better survives the immune response, doesn't trigger it. I looked this up before. I know what it means, but I'm just going to double check. Less immunogenic than normal, and then immunogenic means producing a reaction from the immune system. What's antigenic mean? However, relating to antigen. An antigen is a substance that reduces an immune response, usually foreign. Okay. They may sufficiently by themselves. Okay, they're saying the they here is the molecules. So we put these molecules on the surface of the cells, and it might shield the cells. It might be good enough. Second, in the case of they'll be required to protect against NK cell-mediated missing self-killing. Okay, sure. They're just repeating what they said already, I think. Pretty much. They keep saying the same thing in, like, fancy words. There's definitely been some repetition. I'm not sure there could be a purpose of it. There might be, like, small differences that I'm missing. Like, I can imagine someone reading a philosophy paper and thinking it's repetitive, and I'm like, I was going over, like, multiple points on the same theme, but there were differences that you missed. Like, that could be an answer. So it's possible that's happening here. Third, histocompatibility antigen. The major histocompatibility antigen, 1A and 2 proteins, are the primary drivers of rejection, but mismatch of minor antigens may also play a role. Okay. A number of physiological and pathological states, modulatory molecules occur naturally. They include these ones. Play to greater or lesser extent in fetal maternal immune tolerance, as well as by cancer and certain pathogens. Oh, fetal maternal immune tolerance. So when you're having a kid, you have a bunch of foreign cells in your body in some sense. So your body has to not kill it. As well as by cancer and certain pathogens as immune escape mechanism. These four molecules will emerge as the top contenders. Okay. They have some candidate molecules that you put on the cells, and then maybe the immune system will be okay with them. So you pretend it's your baby, and it might even work in men. Experimental data to date shows that people have tried two things. Class 1B and immune checkpoint. They're saying experimental data shows that engineering approaches, do they mean published? Like, this is vague and poor writing, if not poor thinking. I think they're just saying data shows that what we've tried is two things, which is like a ridiculous thing, and they're dressing it up fancy. Like, you could just say experimenters have tried 2 strategies. Or like a review of published papers found that experimenters have tried 2 strategies. But the mention of engineering approaches isn't like it's not clear that it's referring to what we've tried rather than what we could do. Like, you could be talking about there's like 2 ways to approach this problem in theory, but I think they're just saying we're trying 2 ways right now, experimentally. But they're making it sound like the data like prove something about the approaches rather than the data is just the data of what people tried literally. Like, it's really an odd statement. Okay. Other molecules may also work in the future. Okay, so I think I'm right that they're just saying we're trying these 2 things right now. We haven't figured any other ones out. I don't know how like the 1B and the 1A are related, but whatever. Display minimal polymorphisms. So poly is like many and morph is about change, so occurring in several different forms. The occurrence of different forms among the members of a population or colony. So yeah, you got variety, the presence of genetic variation, occurrence of a number of alternative forms within a selection of nucleic acid or protein molecule. Okay, so you got less variety with these G and E ones. Geared toward immune inhibition rather than activation. Were the other ones not geared to inhibition? I thought they were. So that's what we're talking about. These ones are immune checkpoints. Are these ones listed earlier? Oh, yeah. Okay. Oh, these are the ones they already listed. Oh, I thought they were talking about other molecules, but they went back to talking about the main ones. So they're saying they have 4 things they've been trying. And then they fall into 2 groups because they group the first 2 together and they group the second 2 together. This is related to SENS. I'm trying to figure out if life extension science is any good or not good. Like, are these people maybe going to save my life or probably not? Okay, so they brought up this sentence and they just abruptly switched back to the other ones without really saying they were doing that. But we're now back to the mainstream things, not the other category. So category 1 is these 2, and then we have the other 2, or the immune checkpoint ones, rather than the HLA class 1B ones. Okay, so the immune checkpoints are inhibitory immune pathways. Oh, as an overview, they're talking about stem cells, and they want to be able to make stem cells that are generic so anyone can use them rather than developing them for a specific patient. That way they can mass produce it, and it'll be like way cheaper, and it'll be available in advance instead of like you got sick and then they have to make it, and so on. So it's much better if you can make one-size-fits-all stem cells. And the main problem with one-size-fits-all stem cells is that your body can detect foreign agents. So it says, oh, this is not my cell, this is foreign, I'm going to kill it. So they're talking about how they prevent the body's immune system from killing the stem cells. basically your own cells have an identification marker. So then if you use generic stem cells and then you put it in me, those aren't going to have the identification marker for me, because everyone has like their own personal ID, basically. So if you give me stem cells without the ID, then you need to put molecules on them that prevent them from getting killed. So these are 4 different molecules that might help with that. The immune checkpoints, which are inhibitory immune pathways critical for maintaining self-tolerance, control the extent of immune responses to minimize the killing of bystander healthy cells. I don't know what that means, really. But I know it's not scientific details. Like, this is either they're going to give more details later, or they're relying on you already knowing the details. I can tell for the entire paper so far. Like, if you don't already know what you're talking about, this isn't going to fix it. This paper is not presenting, like, the key facts of science. It's relying on, like, a bunch of assumptions and building on some things people already think to some extent. Although they only have, like, 5 citations so far. So they're, like, a lot of this stuff is general knowledge or something in the field, I guess. Because they're not citing all that much. G is a non-classical. Minimally polymorphic. Okay, so we're now in like a new section. We're just going to talk about G for a while. Like, here's G again. We're still talking about G. Now we talk about E. They just do 3 paragraphs about G with no like section breaks. And then they start talking about E. E gets 1 paragraph, and then the other 2 get 1 paragraph. And then are they going to conclusion? How close are we to universal cells? This seems like sort of a review or overview paper. Like, it's not about any particular science experiment, so this is not ideal. Like, I don't think they're claiming they discovered anything. I think they're just sort of summarizing the state of the literature for some reason. I'm gonna look at their the intro paragraph again now that I've read more. Okay, if we could achieve this, it would be transformative. It'd be a big advance. It would be useful. This paper outlines the principles underlying the project and talks about the advantages of I think this was just terrible writing. I talked about this last time. But anyways, they consider which of the strategies people have already tried are good, and they look at some recently published data. They're not actually doing any science, they're just looking at the field and doing some high-level overview. The citations might have an actual science paper, like experimental science paper. This isn't like a theory paper, really. Like, it's not like, here's my new theory of how this works. Like, it's not trying to invent something, I don't think. It's more like an organizational paper, like, we're not contributing a big new idea, we're just looking at the ideas and trying to organize them a bit better and sort out what's going on. I think that's what it's for. Okay, so I was here, HLAG. I think I'm going to speed up reading because I have a handle on what's going on now. I think it's going to be hard to find a flaw in their science because they're not saying very much. Like, mostly they've said there's this problem of the autoimmune response, and we have some ideas about molecules that might help with it. like one of the molecules that fetuses have. That's broadly reasonable, but it's not saying much. Okay, so they're saying that natural variance in your body or cells or whatever, or genes, that makes you have less of this stuff results in miscarriages, etc. So that's evidence that it helps. And it's unique because it works broadly. And they list a bunch of stuff it works on. Normally it's not used after birth, except in the cornea and a couple like limited cases. So I don't know what an immune escape mechanism is. I wonder if Googling it will actually turn anything up or not. Let's find out. Okay. It's totally a thing you can look up. To some extent. I think they just mean evading the immune system. By losing antigenicity or loss of immunogenicity. Okay. Growing evidence supports G in organ transplant. Okay, so they're saying if you give them G during a transplant, then it helps the transplant success rate. Okay, so they have 3 reports that this G thing might work with stem cells how they want. So they don't have a full explanation of how this stuff works, like in all detail, but they have some like broad general explanations. They're not just looking for random correlations. Like this seems okay. Okay, so 15, 16, 17 would be actual science papers if I wanted to look at them. Uh, okay, so 2 of these papers are from the same group. Nafis, Annalisa, Jim, and Gregory. Okay, so it's different people. Okay, so G seems possibly the best. Now they move on to E. They're really not saying much. There's just some initial research, like 3 reports that seem like maybe promising, worth doing more research. But it's like not that far along. This is the kind of thing where it seems pretty plausible to me that they would get it to work somewhat. Like it would be helpful, it'd be worth using, it would save some people. Um, but getting it to like, very, very consistent, gonna just dramatically increase longevity for almost everyone. Sounds like a lot harder. Like, because you can be a bit sloppy and not totally know what you're doing and have like an 80% success rate. But when you want to get like a 99.999% success rate, you have to be like way more exact and precise and have all the fucking details worked out. And we're sort of blundering around like using things we don't fully understand. Like, we can't just like engineer whatever we want from scratch. We don't have the full documentation of the system. We're taking existing things and trying to figure out which ones might be useful and then where to put them. Which is the kind of thing that can get you like mostly working, but it's really hard to get past that without getting like much more advanced. That's my broad guess. But I would call this not super promising. Like, it's promising in terms of medicine. I think it could help people, but I don't know how promising it is for like I'm going to use this therapy 100 times and it's going to work every time perfectly and it'll keep extending my lifespan a year every time. Like getting at that sort of reliability when they barely know what they're doing and they're just throwing things together that seem like the right things seems really unreliable. So it could still be good, it's just I'm skeptical from what I've read that this kind of thing is going to fit the picture that Aubrey somewhat presents to the world, but not entirely. Like, he's not. Some people want to read him or hear him a certain way that, like, we'll just get these therapies and then they'll work and then they'll protect us and whatever. When I look at this, I'm like, this is not going to be perfect. This is going to help a fair amount of people a fair amount while being quite flawed. So it's not just like, because the way they talk about it sometimes is the initial things will like add 2 years to our lifespan. And then we like get another medicine, it adds like 5 more years. And then during that time we get another medicine and so on. And like that model sort of assumes the medicines work. Whereas this is more like we get a medicine that works like 80% of the time on 80% of the people. And it helps them, but it's just never very reliable. So like the lucky people get saved, but other people don't. And then during that time, we're like losing people to the medicine failing, not just to the clock, while we develop the next medicine while also trying to refine this one. What? Life extension preventing death? I'm not clear what you're talking about. If you just keep preventing death, your life gets longer? Like, you repair a bunch of damage and you don't die of cancer and then you live longer? But what we want is, like, a lot of life extension, like, hopefully, preferably. So to get to, like, a lot, like, makes a huge difference instead of just makes things a bit better. You need tools and components that are kind of reliable, that you can, like, reuse over and over and it keeps working. Okay, so now I have a broad conceptual criticism. Like, it doesn't mean the paper is wrong, but it means the paper as it fits into Sens's desires. It leaves a lot to be desired. Like, it makes... I am now a bit more skeptical when Aubrey says there's like a 50% chance of reaching longevity escape velocity in 25 years or whatever he said. That's not an exact quote. So life extension is primarily about, what I'm talking about is about repairing the damage from aging. So it's not about curing pneumonia or the coronavirus. Those are illnesses or diseases. And medicine addresses those, and they're important to living longer, but it's not the focus of this kind of research. This kind of research is looking at what sort of problems build up over time in the body. So the older you get, the more likely you are to have that problem. And then how do we repair those problems? How do we go in and like clean out the gunk? And if we could periodically do maintenance on the human body and clean out the damage that accumulates over time, then it'd be sort of like staying healthier and younger. That's the idea. And Sens has a list of 7 types of damage. And the last time a new type was discovered was, I think, 1972. So Aubrey came out with a list and no one's been able to add a new type of damage. So he says, maybe this list is actually complete. If we fix these 7 things, then people will live significantly longer. And then when they start living longer, we might run into new problems. But the 7 things might be complete for what we need to fix at current lifespans to get longer lifespans. And not just a little longer, but like significantly longer, because if something would kill you at 110, then due to variance, it would kill some people at like 70. Some people are going to die of it early. So if something is killing no one right now, if we haven't seen it in anyone, or like almost no one, then it probably doesn't kill you until like 250 or more or something. And that's why no one gets even close and never dies of it. So if we fix the 7 things, we might like live until 250 on average. It could be like a big jump like that. But for that to happen, our fixes for each of the 7 things have to be robust so that we can keep doing it over and over until we go from age, 50 to 250. But this stuff they're talking about is not currently robust enough, like anywhere near robust enough for that to work. Like we might get a situation where it's like, if this therapy works 200 times in a row, you can live to 250 years old. But the chance of it working 200 times for you is like low because it's not reliable. Now, maybe when it doesn't work, you just try again. So maybe it's not a big deal, but maybe when it doesn't work, bad things happen. I do not know. It also depends how expensive it is. Like, because for retries to be okay, you want it to be like harmless and cheap when it doesn't work. And then it wouldn't be a big deal. You can have even like 50% reliability. It's not going to be a big problem because you can just retry a bunch of times. But if it's expensive or harmful when it fails, then retrying a bunch is going to be really bad. Oh, and the other thing I was going to say is, so cancer is not an illness, or it's not a disease. Aubrey has talked about this. It's a pretty important distinction. Cancer should not be compared with AIDS, pneumonia, flu, cholera. It's not like bacteria or viruses. It's not an acute, like an infection. Cancer is a different sort of process that we're not going to cure like we have a cure for smallpox. Instead, cancer is essentially, it's part of the aging buildup damage category rather than the disease category. So it's one of the things SENS works on is cancer. Okay, so they're saying you put the G on, and then it can knock out the B cells and the NK cells and the macrophages. Or that, no, that's inhibit. It inhibits those. It induces Treg cells, whatever those are. And then the E one over here inhibits the NK cells and the CD8 T cells, but it doesn't inhibit monocytes, dendrites, macrophages, B cells, etc. And then up here, they have the CD47 and the PDL1, which I haven't read about what they do yet. But apparently this one inhibits macrophages, and this one inhibits CD8 T cells. And then over here, there's something related to NK cells. And then this one isn't even connected to something, but... Oh, this is you can knock out the class 2, and this is you can knock out the class 1, which induces killer cells to come kill you. Right. Okay. Okay, I understand their diagram now. This doesn't act, it's not actually that like fancy or complicated once you have an idea of what's going on. Like the details of how it works are fancy and complicated, with the amount of information in this diagram, I think I understand now, basically. Okay, that's where I was. I need to scroll down. All right, now the CD47 and the PDL1, how do these work? I didn't really follow what they meant by checkpoint. CD47 is on all human cells, plays a role in self-recognition. It's a don't eat me signal to macrophages. So CD47 up here is the one linked to macrophages. And it inhibits them. Inhibition. Achieved through that. There's a receptor. Extensively upregulated in solid and hematological malignancies. I actually know this word. Okay, hematological is like blood related. The study of the physiology of the blood. Hematology. Yeah, blood related. Okay, I knew that word too. Branch of biology that deals with normal physiology, right? Normal functioning of living and organisms and their parts. The way in which an organism or bodily part functions. Okay. The number of anti-CD47 monoclonal antibodies are in human trials as a cancer immunotherapy. So I think what they're saying is you destroy the CD47 on the cancer cells, and then your immune system kills the cancer. I think that's what they're saying they're trying to do. A number of is bad writing in general. You can just say some. They're trying to be fancy, but it's like stupid and really vague and generic. Like, it's a number. That's like poor info if you think about it literally what they're saying. It is some number. Like 0 is a number, 1's a number, 1,000,000 is a number. It's as vague as you can get. If you say like a few or several or something, it's more specific. It would be better. People usually use a number of for basically several. They're talking about like 3 to 6 or something. Maybe a few more. Why don't they just write like approximately 5 or something? It'd be better to give more info. Alright, now we're switching. So PDL1, also known as CD274 or B7H1. Okay. Expressed physiologically. Some of the things they say are weird. Like, if this paper is for experts, don't they already know this? Why are we telling them this? Like, the paper relies on you already knowing so much stuff in advance, and then they tell us the alternative names of something. Like, why is that the info that their target audience needs? I don't understand. It'd be like if there's a philosophy paper that was written so a layman could not understand it. It was constantly talking about like advanced stuff. And then they also throw in that critical rationalism is also known as Poppurian epistemology. Like, that is not useful info for an expert. expressed mainly on placental tissue. And pathologically on cancer cells. Pathologically means it's bad. Related to pathology or disease. Pathology means disease, malfunction. They don't define this very well, I don't think, but yeah, this is better. We just go to the old dictionary. The science which treats diseases and their nature, causes, etc. Okay, anyways. So PD-L1 delivers a don't find me signal to T cells, whereby it binds to the PD-1 receptor located on T cells to inhibit them. And this is meant to help with cancers. So with this one, you can get T cells to attack the cancer if you can get this to affect cancer cells. So these two are like very incomplete. Okay, click on the book right here, Ending Aging by Aubrey de Grey. That book explains the 7 things. But you can also like click on the playlist of 200 videos. He talks about it in like a lot of them, so you'll probably run into it. I had a long discussion with him about philosophy. It was approximately a 210-page discussion. And a lot of it was epistemology. We talked about cryonics some as well. Okay. Anyway, so these two are like nowhere near gonna do what we need. This one does 2 major things. So it's like better. And then this one does like a lot of things, so it's more hopeful. These two, if you did both of them plus this one, then you're between those 3 things. Maybe it's hopeful. Okay, so they're saying we're gonna need 1 or more of 3 engineering strategies. Delete the identification molecules. Use the first 2 of their 4 promising things, or use the second 2 of their 4 promising things. So they're saying that they're pretty sure we're going to use this, those two. They're saying we're going to use something from at least one of these 3 groups. That is, we'll use some of the anythings we've figured out so far. So, not much of a statement. We've got 6 ideas, and if you divide them into 3 groups, then we're pretty sure we'll use stuff from at least one of the groups. These are the main strategies that have been explored so far, although other things might work in the future when people learn more. So it's not at all clear that we'll use one of these three strategies. They're just the first three that we came up with. We might totally figure out something else different and better later, and then use none of these. So it is saying it's clear is just wrong. So there's tons of errors, but it's not like a hard science error. But everything they've said about science is so vague that I wouldn't expect hard science errors. But I did come up with my one main argument about why this doesn't look great. How close are we? What next? I'm getting tired. I think I might have to. I might stop. I don't know if I want to read the rest of this. There's not that much, but it's very hard to read. And I already wrote like 2,500 words earlier and did a few other things. Comment on which thing? I did comment on stuff you said? Yes, there are not economic incentives for short-termism, but some people think there are. So people act that way, but they're wrong. Oh fuck. It spells his name wrong. Is that an autocomplete type of error or is that yeah. Fuck, I'm gonna fix that. Just comment that comment. There we go. I don't like spelling his name wrong. I have I always spelled the word gray wrong, so I made a fix for it. But then it causes me to spell Aubrey's name wrong. See, I spelled judgment and gray wrong, so I made these text shortcuts to remove the E from judgment and to spell gray with an A. E is like the UK version of gray, I think. Yeah, see, British is with an E, but American is with an A, so I should spell it with an A. So my habit is E. Judgment might be the same, I'm not sure. Let's see. Judge. Also judgment, yeah. I think that's the less standard one, at least in America, is to use that E. Like some of the dictionaries list judgment with an E as an alternative. and some don't even mention it. Oh, first place in Yu-Gi-Oh? Oh, I see. Top 8. Got it. Okay, where's my stream one? First of all, I'm gonna edit in the link on the. Okay, keeper right. Okay, and then stream 2 is the save video link. Fill the junk. Okay. Then title. So top 0.5% of people who showed up to an IRL tournament. What percentage of people travel for that versus their like local? Alright, well, later.